Good morning, everyone. I'm here to talk about automated chemical analyzer, or otherwise known as a chemical analyzer or auto analyzer. First of all, myself, P. Bonureka. These are the content we are going to see in this PPT. First is definition, advantage, basic approaches, auto analyzers, immuno-based analyzers, miscellaneous analyzers. Definition. An automated analyzer is a medical laboratory instrument or a device that performs chemical analysis automatically. These measured properties of blood and other fluids may be useful in the diagnosis of disease. Advantages. The human factor is decreased during the mechanical and repetitive part of an assay. Accuracy is more when compared with manual method. A large number of samples may be processed in a minimal time. At the same time, a very small amount of samples and reagents subsequently, allowing less blood to be drawn from each patient. Two or more assays may be performed simultaneously, and automation eliminates the potential errors of manual analysis. The quality of patient test results is monitored continuously for improvement of testing process. The basic approaches is continuous flow analysis, centrifugal analysis, discrete auto analysis, and dry chemical analysis. Let's see brief about the auto analysis. The approaches used in the auto analysis is continuous flow analysis or segmented flow analysis. The overall procedure or working mechanism of auto analyzer, the samples and reagents are passed sequentially through the same analytical pathway and separated by the air bubble. The quantity of sample and reagent is controlled by proportioning pump. The mixing of sample and reagent occurs when two pathways joined form a common pathway. And the different part of the systems and the functions are used in the auto analysis. The main components of the auto analysis is samples, proportioning pump, air bubbles, diluent, reagent, dialyzer, heater, colorimetry, and recorder. This is a block diagram of automated chemical analysis. The requirements of components here, the sample, proportioning pump, as I say before, these are the components we use in the automated chemical analysis. Let's briefly about each component. First is sampler. Sampler, it holds the sample. Proportioning pump, it regulates or controls the quantity of sample, reagent, air bubble and dilute which should be passed to the dialyzer. Air bubbles used to separate the sample and reagent with each other. Diluent used for dilution of sample. Reagent used to form a color complex with sample. Dialyzer, it consists of a semi-permeable membrane which is used to separate out protein-free filtrate from a sample. And next is heating bath. The reagent and the sample react with each other to form a color complex. The formation of color complex requires a specified temperature for a specific time which is provided by the heater. The heater provides incubation conditions for a formation of color complex with the temperatures of 37 degrees Celsius. The next is a colorimeter. It's an intensity of a color complex. It is a major one in a automated chemical analysis. Last but not least is a recorder. It shows the result or a concentration of a component in a graphical form, such as a creatinine, protein, and glucose. The use of automated chemical analysis is used to calculate the concentration of a certain substance within a samples of serum, plasma, urine, and or other body's fluids. The substance analyzed through these instruments include certain metabolites, electrolytes, proteins, and drugs. And the purpose of auto analysis to measure the sodium, potassium, chloride, glucose, and various enzymes. This is a real automated chemical analyzer device or instrument. Next is immuno-based analysis. Antibodies are used by some analyzers to detect many substances by immunoassay and other reactions that employs the use of antibody antigen reactions. In auto analysis, we use samples and reagents. In immuno-based analysis, we use the antibodies. That's a main point in immuno-based analysis. Next is when concentration of this compound is too low to cause a measurable increase in turbidity when bound to antibody. More specialized methods must be used. Recent developments include automation for the immuno nematology lab, also known as the transfusions lab. Next is a miscellaneous analysis. Samples are more in a more performed simultaneously in automatic chemical analysis as the same as the antigen in immunobased analysis, but miscellaneous analysis only a few tests or even for only one test to done and at a time. So the following two categories, some testers saw DNA labeling and detection, osmolarity and osmolarity measurement of glylated hemoglobin, hemoglobin A1C, and aliquoting and treating of samples throughout the laboratory. These are the categories involved in the miscellaneous analysis, only few tests or even for only one test. We cross about in this PPT is auto analysis and immuno-based analysis and miscellaneous analysis. In auto analysis, we see the definitions and components, overall procedures, mechanism, the block diagram, and uses. These are we cross over the auto analysis. In immunoanalysis, we see just only the what the antibodies functions in the immuno-based analysis. And miscellaneous analysis, we only see about the list of a few process done by the analyzers. That's all in this PPT. So that's all. Thank you, friends.